So in this chapter, we are getting started on all of the bacteria that we're going to go over for this course. Again, there are more bacteria out there than what we're going to talk about. We are talking about some of the top bacteria that cause human disease. And we split it up into 4 different chapters because there's a lot of bacteria out there. So this chapter is all about the medically important, so the most common, most likely to cause disease in humans, gram-positive and gram-negative cocci-shaped bacteria. So everything in chapter 18 all has the exact same circular shape. Most of them are going to be gram-positive in this chapter, and then a few are going to be gram-negative cocci shape. So we're going to start with our gram-positive cocci bacteria because there are more of them than gram-negative cocci shaped. And one of the first most common genus out there that causes human disease is Staphylococci. Now, some general characteristics of Staphylococci, we find them a lot on the skin and the mucous membranes. So they're just normally there. There's lots of Staphylococci bacteria. They don't all cause disease. Some of them are part of our normal flora. They are all gram-positive. They don't have any flagella. They don't have any kind of spores. A few may have capsules, and there's 31 species. We're only going to talk about a couple, the ones that most often cause disease. So when we gram stain these, they are gram-positive, so they're going to show up purple after a gram stain, and they have the cocci shape. They generally like to hang out in clusters, so they kind of look like little clusters of grapes when they like to hang out. Now again, there are lots of different gram-positive bacteria out there, cocci-shaped and some of the gram-negative cocci-shape. We're going to look at these tables are going to show this slide and the next. Some of these bacteria we're going to talk about in this PowerPoint. We're going to talk about a Staphylococcus aureus, some other coag-negative staph, Streptococcus bacteria, and again, there's multiple species of Streptococcus that cause disease, pyogenes, agalactiae. We're going to talk about Enterococci. It even tells you the name of its shape right in of its name. Most of these do. It's the Staphylococcus, Streptococcus. So like they tell you they're cocci shaped. Not all of them do that. When we get into the gram negatives, the Neisseria, there's 2 Neisseria that are both gram negative cocci shaped bacteria that cause disease. So of the Staphylococcus bacteria, again 31 species, the one that causes the most issues for us is Staphylococcus aureus. When we grow it on a plate, it's nice, big, round, kind of cream-colored colonies. It grows best at body temperatures. The interesting part, a lot of Staphylococcus, including this one, can withstand a lot of salt, which makes sense. A lot of these Staphylococcus bacteria are found on our skin. Well, there's a lot of salt on there, especially anytime you sweat. There's a lot of salt, and they don't love the salt, but they can withstand that high salt content area, whereas a lot of other bacteria wouldn't. They can change or withstand extreme pH changes and even high temperatures. And a lot of them, especially this one, has a lot of different virulence factors. Now, when we talk about virulence factors, these are things that allow the bacteria to cause disease, so things that are good for it, which then means bad for us. So we're going to look at some of these virulence factors of just Staph aureus. Again, this is the bacteria that causes a lot of issues of the staph bacteria because it has all these virulence factors. And a lot of the virulence factors are enzymes. If you notice, a whole bunch of them end in ace because they're all enzymes, and a whole bunch of toxins. So this one kind of bacteria has lots of enzymes that cause issues, all good things for it, which means all bad things for us, and it has lots of toxins. So just a few of them, it makes an enzyme called coagulase that will coagulate or thicken blood plasma. Now, for it, that's beneficial because it can actually hide in some of these blood clots. It has an enzyme hyaluronidase that breaks apart tissue. It digests tissue. It allows the bacteria to spread to new areas. It has an enzyme called staphylokinase that then digests blood clots. So it can make a blood clot, hide in it, and then break it down. It makes an enzyme called lipase that breaks apart oils, which there's oils found on your skin, so it allows them to easily colonize your skin. It makes an enzyme called penicillinase, which means it breaks apart penicillin. So you're not going to treat a Staph aureus infection with penicillin. It also makes toxins. Hemolysin break apart red blood cells. And again, a lot of these toxins lead to a lot of the issues that come from, and a lot of the diseases and disorders that Staph aureus cause. It's got a leukocidin that breaks apart white blood cells, enterotoxins that usually cause some type of food poisoning type symptoms, exfoliative toxins, which actually cause the shedding of our skin, and then a toxic shock syndrome toxin, which causes fever, rash, blood pressure issues, organ damage, all bad things. All good for the bacteria, because it allows, a lot of these things allow the bacteria to get to different places in the body. Now, where we find Staph aureus? Well, it's pretty much anywhere humans are. It can easily be found on lots of different fomites. Those are inanimate objects, so doorknobs, tables, and I'm like, pretty much anything you look around right now, wherever you're listening to this or watching this, and I'm like, it's probably going to have some staph bacteria on it, including possibly Staph aureus. The carrier rate for Staph aureus, between 20 and 60%. Now, that's a big range. That means 20 to 60% of you guys carry this bacteria as part of your normal flora. But it's got a lot of things that can cause disease. Yes, it does. But a lot of times, if you have a healthy immune system, and if this bacteria doesn't go somewhere where it's not supposed to be, like skin, mucous membranes, so if you're healthy, you're generally okay. If you're immunocompromised, and this gets to somewhere where it's not supposed to be, that's an issue. Because guess who you're working with in the healthcare facility? You're working with those immunocompromised patients. And a lot of them have open wounds. And you might be a carrier of this bacteria. Now, 20 to 60%, big range. Usually, the higher the number, that higher percentage, is for those that work in healthcare. So, yay, if you already work in healthcare, you are highly likely to be a carrier of Staph aureus. It really likes to hang out in your nose. It really likes to hang out in your skin, your nasopharynx, and it can also be found in the intestine. So this is one. Again, I'm a little sad that we're not doing, that this is an online versus face-to-face, because if this was a face-to-face lab, you would actually be swabbing your nose and we would be testing you for this particular bacteria. Otherwise, when you go into the healthcare field, they do a lot of these tests to find out if a healthcare worker is a carrier of this bacteria. Again, it's just that you're aware to make sure you're taking all the proper precautions so that the bacteria doesn't get into open wounds when you're working with patients. Otherwise, those that are at a highest risk of then picking it up, that poor hygiene, it's the immunocompromised part. If you've got other existing infections, diabetes, tissue damage, nutritional issues. And the problem is this particular bacteria is becoming resistant to antibiotics. Methicillin was the top antibiotic we used to prescribe for Staph aureus infections, and like a lot of other bacteria, it is becoming resistant now. So because of that, the strain of Staph aureus that we say is resistant to the antibiotic methicillin, we give it a fun little nickname, MRSA, and it stands for methicillin-resistant Staphylococcus aureus, meaning it's the Staph aureus strain that's resistant to methicillin. Not all of them are resistant. Now, this is just to show you that, yes, the bacteria can get into the skin, into the follicles. It can grow. It can cause infections around hair follicles. Again, we can sometimes see that in infection of a hair follicles, folliculitis. It becomes inflamed. Now, if it becomes inflamed and not, that's usually when you start to get abscesses, and it's harder to treat. It's methicillin resistant if it's the strain. Your body can't get rid of it on its own. Or at least not for a longer period of time, and it's just harder to treat with different types of drugs. Otherwise, some of the other diseases that it can cause, again, kind of depending on where it's at in the body and the health of the immune system, it can cause that folliculitis. It can cause something known as a furuncle. That's inflammation of your hair follicle with an abscess or a pustule from there. A carbuncle is kind of like a really bad furuncle, which I just like the names. So it's a larger, deeper lesion. Impetigo is when you can have some kind of swellings on the skin that start to peel away. It can also get into other parts of the body and cause systemic infections, meaning it affects other body systems. It can get into the bone tissue, cause something known as osteomyelitis. And when it gets into bone tissue, it can get into that bloodstream, and it can be traveling anywhere throughout the body. When you've got bacteria traveling anywhere throughout the body, you have now bacteremia. You've got bacteria in different parts of the body that can pretty much go anywhere. Now, if it gets to the heart, it can cause more serious side effects. Now, some other things that it can cause food intoxication. If you eat something that has this particular bacteria in it, so you're eating the toxins that it makes, it can give you food poisoning symptoms, the diarrhea, the nausea, the vomiting. One of the toxins that this bacteria makes, it causes the skin to slough off. So it's called scalded skin syndrome. It almost looks like your skin got burnt, like a 1st or 2nd degree burn, and then it starts to slough off, kind of like after you have a really bad sunburn. But it can get into the bloodstream, and it can travel anywhere. And because of the toxins that it does have, it can cause damage to various types of organs and even organ failure, and it leads to the condition known as toxic shock syndrome. Now, when you may have heard of toxic shock syndrome, there are other bacteria that can cause it, but one of the top ways to get it, it's because of the toxins, and one of the big ways to get it, the most common ways, are tampons. Is because you're putting a foreign object that into the body that's going to stay there for a long period of time, and if there are any bacteria whatsoever that are on that tampon, you just gave it the perfect environment, 37 degrees Celsius, it loves body temperature. I'm like, you just gave it a place to grow. So that's usually, it's like the most common way that people end up picking up this particular bacteria and leading to the toxic shock syndrome. Now, this is just showing a couple of the other diseases, some of the kind of, we're getting into the carbuncle furuncle stage and the scalded skin syndrome. Now, some other important staph, because again, 31 species. Staph aureus is the one that usually causes the most problems. And one of the enzymes that Staph aureus have is an enzyme called coagulase. It can cause blood clots. Well, there's other Staphylococcus bacteria that don't have the enzyme coagulase. So it's like 30 bacteria don't have coagulase and one that Staph aureus does. So we're going to talk about just a couple of the other staph bacteria that don't make that enzyme coagulase. Now, 2 of them, and only one I'm really going to talk about. The one is Staph, S. epidermidis. Based on its name, it lives on the skin. It's found in the mucous membranes. However, if it gets into the body because you have some type of open wound, someone wasn't wearing proper PPE and their skin bacteria got into an open wound, it can travel throughout the body. It can cause endocarditis. It can cause bacteremia. It can get into the urinary tract and cause UTIs. So it's usually higher immunocompromised individuals have a higher chance of developing issues, and if it gets somewhere where it's not supposed to be, supposed to go. Staphylococcus hominis, this is the one that lives around the sweat glands. It likes that high salt. It likes the kind of the proteins that come with the apocrine sweat, and it grows. It's one of the bacteria that does make you smelly when you sweat. Now, trying to identify, again, there's lots of staph out there. We're only talking about some of the top ones that cause issues for humans. But how to identify them? Well, we can find staph in a lot of different areas, but we can grow them up in a lab on plates. And one of the first things we're going to do is test to see if it is catalase positive or not. All Staphylococcus bacteria make an enzyme called catalase. I'm actually going to flip to this next slide, which talks about the catalase test. So all staph bacteria make an enzyme called catalase. Now, what that does, it's an enzyme that breaks apart hydrogen peroxide, which is a toxic form of oxygen. When it breaks apart hydrogen peroxide, it actually releases the hydrogen gas as well as an oxygen gas, which means you're going to see it as bubbles. So we can add hydrogen peroxide right to a little bacteria on a plate. We can put it on a slide, and we're going to look to see if it's bubbles. If we see bubbles, we know it's a staph bacteria. And it means we don't know it's not one of the other bacteria we're going to talk about in this PowerPoint. We know it's a staph. Then we can start doing another test. We can look to see if it has that enzyme coagulase. Now, coagulase means clotting. And so in a lab, in a tube, we can look and see if a clot forms in some blood plasma and some blood serum. If it does, it's because the enzyme coagulase is present and we know we have Staph aureus. If it doesn't clot, if it doesn't clump up, we know we have some other Staphylococcus bacteria, one of these coag-negative, 30 of them coag-negative Staphylococcus bacteria. So we use a catalase test to tell us that it's a staph, and we use a coagulase test to start to narrow down what kind of staph. Now, because there's a lot of coagulase negative staph bacteria, we usually have to run some other types of biochemical tests. We can run and test a whole bunch of different enzymes, sugar, breakdown, and based on color changes, we can determine what the bacteria is. Now, there is a flow chart in your book, table 18.2, that kind of goes through some of these different things that we look at to help ID different types of staph. Whether it has that coagulase is it coagulase positive? Does it have that enzyme or not? Is it anaerobic? Can it grow in an anaerobic area? Can it break down an enzyme urease? We're just looking at different tests, different ways to figure out which staph bacteria it is. Now, the clinical concerns we run into with staph bacteria is that a whole bunch of staph, do make that enzyme called penicillinase. And it just means they're resistant to penicillin and its closely related antibiotic ampicillin, which means you can't use those to treat for a staph infection. And then some we used to then treat with methicillin. We now have strains, our MRSA strain, that is methicillin resistant Staph aureus. So we're getting a lot of staph bacteria that are becoming resistant to a lot of our antibiotics. Otherwise, other treatments, like if there's any abscess, you're going to have it surgically removed. They don't want you doing any of this at homes because it could be spreading that resistant strain of bacteria to other areas. If it's a systemic infection, if it's spreading throughout the body, it's generally going to require intensive, lengthy therapy, strong antibiotics, usually in vitro antibiotics that it's going to be hard because a lot of people can and do die of staph infections, especially once they get into the bloodstream. Now, our MRSA is not the only resistant staph bacteria. Again, Staph aureus, we do have things to treat Staph aureus. However, if you have the methicillin resistant strain, we just have different antibiotics to treat. And then now we have a couple more resistant strains. We've got vancomycin resistant or vancomycin intermediate. It has little effect, but some effect. And that these are some of our last drugs that we take. Staph is becoming very resistant to antibiotics. And the more we use, the more they get resistant to it. Now, preventing from getting a staph infection, because it'd be best if we just didn't have one whatsoever. Then we don't have to worry about it. Well, universal precautions by all healthcare providers, because a lot of healthcare workers carry this in their nose as part of the normal flora. So a lot of times the healthcare setting is going to test you to see if you're a carrier or not, and you might be barred from working in nurses, operating rooms, or delivery rooms where you might have high-risk patients and open wounds where that bacteria could get in. Otherwise, it's generally mask over the nose, possibly face guard, anything to prevent any of your bacteria from getting into open wounds. Carriers can be treated. So if you came back as being a carrier, you could be treated with a combination of a bunch of different drugs to get rid of the bacteria. Problem is, you are more likely to pick it up in the future. Otherwise, it's good hygiene, making sure you're washing your hands all the time, making sure all surfaces are clean, and they are working on a vaccine to provide protection against this particular bacteria. Now, my first little concept check I have on here, which test differentiates Staph aureus from other Staphylococcus species? And I'm like, and it is the coagulase test. So, gram stain, all staph bacteria are going to look the same. They're all gram-positive, they're all purple, they're all cocci-shaped, and they all like to hang out in little bunches. All Staphylococcus are catalase positive. So that doesn't tell us which staph, it just tells us it is the staph. And then oxidase we haven't gotten to yet, but Staph aureus does make that enzyme coagulase, which would form a clot in a tube in a lab. So this is kind of my little, if we're trying to identify different gram-positive bacteria, a gram-positive cocci, if you gram-stained and you saw purple circles, you got gram-positive cocci. You could do a catalase. If it's positive, you know it's a staph. You do a coagulase to know, which staph. Staph aureus is always coagulase positive. Staph epidermidis, it's a coag neg, and it's kind of one of the most likely staph infections. If, however, you did a gram stain, you had gram-positive cocci, and it showed up as a catalase negative, then you're in this category here. It's either going to be a Streptococcus bacteria or an Enterococcus bacteria. And yes, based on their names, these are all cocci-shaped bacteria. So we're going to take a look at the Streptococcus bacteria. So some just general characteristics of strep bacteria. They're gram-positive. They are cocci-shaped, although some can get a little oval-ish depending on the different strep bacteria. A lot of them like to hang out in chains and in pairs. They don't make spores. Some can form capsules. Some can make slime layers. Some are anaerobes. They do not make that enzyme catalase, because catalase tells you it's a staph. Most are fastidious, means they're kind of the picky eater bacteria. They really like red blood cells, but they like them broken down for them first, so we have to cook them for them. So they require a more picky media to grow on. When we do grow them, the colonies are really small, they're non-pigmented, they don't have any particular color, it's usually kind of a white cream. And this bacteria are sensitive to drying, to heat, and disinfectants, which means they usually don't stay on surfaces for a very long period of time, and they're more easily to get rid of. This is just showing how you can see some of them forming those nice long chains together. Now, because there are lots of strep bacteria, we group them based on a structure on their outer cell wall. And so we put them into a grouping called a Landsfield classification or a Landsfield group. Now, generally the ones that they're just little antigens on their cell wall, the ones that generally cause the humans most issues, so they're the ones we care about, are ones that have A antigens, B antigens, some C, some D, but A and B are the top antigens on the cell walls that cause human disease. Another way to sort out and group all of the Streptococcus bacteria out there is we're going to look at something called hemolysis, which means, lysis means break apart, heme means blood, so it's how does the bacteria break apart red blood cells. So in a lab we can put bacteria on plates that have red blood cells in the media, and we can look to see can it or how much can it break those red blood cells down. So we break it into, if they can break the red blood cells down, we call them either Beta-Hemolytic, which means they can completely get rid of the blood cells to the point you could see right through the media. If it's Alpha-Hemolytic, it's a partial breakdown, which means we can see the red blood cells aren't fully there anymore, but instead we know there's some breakdown, and it usually actually leaves kind of a greenish pigment behind. So we can't see through the plate, but we know the red blood cells are breaking down. So we're using these antigens on the cell walls and hemolytic reactions to help determine what kind of strep it is. So this is showing a beta hemolysis, a complete clearing. You can actually see through, this is the colony itself, you can actually see through the media around where that colony is. On a more red plate, they drew out with a beta hemolytic bacteria. They were so clever, and all the red blood cells are gone anywhere the bacteria is. Alpha hemolytic It means there's some breakdown. This is not my favorite picture. There's some breakdown, but you can't see through the media. I like this one. Again, they use an alpha-hemolytic bacteria to draw the little alpha symbol, and it does kind of have a more greenish pigment. So we know there's some change, some breakdown of red blood cells, but it's not a full breakdown. Another, if you're wondering what the third one on my little plate there is, it looks kind of like a Y. It stands for gamma. It's a Greek symbol for gamma, and it just means no breakdown, which means if I looked at this plate, held it up, I could flip the plate over and it would just look as bright red. You'd see every single red blood cell there. So, some of the bacteria we're going to talk about in the streptococcal group. Again, there are some beta-hemolytic, and there's more than one beta-hemolytic. We group them based on their antigens that they have, A antigens, B antigens. We also try to figure out what are they based on if they're sensitive, meaning affected by, or resistant to certain antibiotics. And so there are some bacteria that are naturally resistant or naturally sensitive to certain antibiotics. And knowing that, it helps us figure out which bacteria it is. So we've got some beta-hemolytic bacteria. We're going to talk about strep pyogenes. Alpha-hemolytic bacteria. We're going to talk about the strep pneumoniae, as well as some of those that are in what's called the Viridans group. Again, we can sort them out based on their antibiotic sensitivity. So these main ones in bold, again, we're not going to talk about all the strep, all the enterococcus, but we're going to talk about some of these in bold. The strep pyogenes, the agalactiae, the Enterococcus faecalis, as well as the Streptococcus pneumoniae. Again, lots of bacteria out there. Not going to talk about them all. So, Streptococcus pyogenes. Out of all the strep, it's the most serious streptococcal pathogen. And just kind of like staph, it's because it has a lot of virulence factors. Now, this bacteria usually likes to hang out in the throat, the nasopharynx, and occasionally causes skin infections as well. Now, some of its virulence factors. Yeah, there's 3 sides of them. It does have some surface antigens. It has a carbohydrate called the C carbohydrate that protects against lysozymes, so our own cells can't break it down. It has fimbriae that allows it to attach to surfaces. It has a protein called the M protein that resists phagocytosis, so it doesn't even get eaten. You know, the one part of your immune system is eat bacteria, it prevents it. It has a hyaluronic acid capsule that hides it. It provokes no immune response. It provides a little camouflage. You can't eat something you can't see. It has another enzyme called C5 or complement protein, C5a protease. It hinders or stops complement proteins and neutrophil response, which means you're not going to cause lysine of cells. You're going to stop white blood cells from going after you. This is showing on the outside that it has some of these enzymes and various antigens just right there on the surface of the cell. But otherwise, other than different enzymes and other surface things, it has toxins as well. It makes a toxin called streptolysin that breaks apart different types of red blood cells, which allows us to figure out what kind of strep it is. And this strep pyogenes can break apart red blood cells fully. It makes a toxin called erythrogenic. Anything erythro means red. Pyro means heat, so it's a pyrogenic toxin. It induces fever and a red rash, as shown on the picture. It also makes enzymes, streptokinase, to break apart different types of clots that allows this bacteria to get to new surfaces and new areas in the body. It makes hyaluronidase, which, just like Staph aureus does, it allows this bacteria to break apart different connective tissues and spread to different areas. And it makes an enzyme called DNase, which breaks apart DNA, which is found in all cells in our body. It helps break apart cells and get to new areas. It helps break apart pus. And so it can also get to new areas by kind of having a more liquidy type pus. Now, where staph pyogeny is found? Well, humans luckily are the only reservoirs. You can't get this bacteria from any other pet or anything else. Strep pyogenes, because it does like to be found in the mouth, it is the bacteria that causes strep throat. The strep is short for Streptococcus, but there's lots of strep out there. But Streptococcus pyogenes is the bacteria that causes strep throat. It's transmitted by skin-to-skin contact. Kissing's going to be a big one. Any kind of water droplets, talking, and just kind of spitting out when you're talking. It can be transmitted on food or fomites, the inanimate objects. Portal of entry, it normally gets in through the skin or in through the pharynx, breathed in. Children are the predominant group that are affected for cutaneous and throat infections. Lots of kids get skin infections from this bacteria. Lots of kids get the strep throat from it. However, it is a bacteria that can leave the skin and it can leave the throat, and it can cause some very serious issues afterwards. So if left untreated, it can cause a lot of other issues throughout the body. So, some skin infections. Well, it can cause something known as impetigo or pyoderma, is you've got lesions that are going to form all over the skin. It is highly contagious, and so in daycares and kids that have it, anytime they're touching skin, it could be spreading. You're spreading the bacteria. Erysipelas, this is when it gets into broken skin, so any type of scratch, abrasion, anywhere, and it can spread around inside the dermis and the subcutaneous area, causing kind of these lesions on the skin. Again, it is the bacteria that causes strep throat, which is more officially called streptococcal pharyngitis, inflammation of the pharynx caused by the Streptococcus bacteria. Now, big sign of it is back. At the back of your throat, you're usually going to have very red, inflamed tissue, and a lot of times you're going to have these pussy patches as well. However, if the bacteria gets into the bloodstream, it can go to different places. Because it has toxins, it can cause a condition known as scarlet fever. Now, it's because that toxin, the scarlet is because, yes, your skin turns very, very red. However, that toxin, other than causing a red tissue, can actually cause damage to other areas in the body as well. It can cruise around in your body and your bloodstream, causing septicemia. It produces a toxin that can lead to toxic shock syndrome, except we call it streptococcal toxic shock syndrome, just so you know that it's not a staph toxic shock syndrome. But it's also a bacteria with the toxins that it creates and the enzymes that it makes. Those enzymes and toxins can break apart tissue. That's great for the bacteria because it allows the bacteria to get to new places. However, it can cause a really serious, deadly disease called necrotizing fasciitis. You may know it as the flesh-eating disease, that this bacteria eats apart your flesh. And it's trying to get to new areas in the body to inhabit, but the problem is you can't grow that flesh back, so it usually is going to require amputation to stop the travel of the bacteria to new areas. Now, if there was left untreated scarlet fever, that toxin is throughout the whole body, causing that red skin, it can lead to something known as rheumatic fever. Not always, but it can. And rheumatic fever, it follows untreated strep, untreated scarlet fever, and it can damage heart valves. Now, reason why I have the picture on the bottom of Bill Paxton, if you didn't know, the actor, he actually died a few years ago. He had suffered from rheumatic fever as a kid, and he had known that. There are some, audios of him talking about it. Yes, I had rheumatic fever as a kid. I know I had some heart valve damage. Well, he went into surgery to have those valves replaced, and he died from complications that happened afterwards. So rheumatic fever, because of all the toxins, depending on where the toxins go, depending on where various destructive enzymes go, it can damage heart valves. Now, the toxins can also damage the kidneys, and so it can cause a condition known as acute glomerular nephritis. It can, if those toxins stay there long enough, actually cause kidney failure. Now, this is just showing how it can cause some of the damage to the valves, that the valves themselves might not be able to close properly, meaning depending on which valves, you could have backflow of blood or blood not getting to different parts of the body where needed. Now, another concept check. Which of the following is not a possible complication of strep throat? Well, strep throat, if left untreated, it can cause the scarlet fever. That's you're going to get red everywhere. It can then cause rheumatic fever. This is, that next step where it's going to damage the heart valves. And then the acute glomerular nephritis is damage to the kidneys. So the only one that generally is not an issue with strep throat is pneumonia. Now, another strep bacteria. This one has B antigens on them for the Lancefield classification. It's Streptococcus agalactiae. Now, this is the bacteria that's normally a concern with pregnant women and newborns, is because this is a bacteria that can be part of the normal flora in the human vagina. It can be found in the pharynx and large intestine as well, but the issue becomes if it's part of mom's normal flora, this is a bacteria that can get transferred to an infant during delivery, and it can cause a lot of serious infections in newborns. Again, they don't have a really strong immune system. It can cause the newborn pneumonia, sepsis, meningitis. So it is a bacteria that pregnant women are screened for. They do swab them closer to childbirth time in their pregnancy, and they look to see if they're a carrier of this bacteria. If they are, they do give them antibiotics to get rid of the bacteria before childbirth. Otherwise, the bacteria can get into different kinds of wounds and skin infections. Depending where it gets in the body, it can cause endocarditis. That's usually more for those that are of a weaker immune system. Now, we also have a couple other, strep and enterococci bacteria. So there is a group of bacteria, they're enterococci. They're called entero because anything enter or entero has to do with the intestine. And you'll see entero in a lot of words when we're naming a lot of different organisms. So entero has to mean intestines. So Enterococcus faecalis, Enterococcus faecium, Enterococcus durans. These are all normal flora of the large intestine. They all have group D antigens on them, and depending on where they go, so normal flora of the large intestine, but if they get somewhere where they're not supposed to be, they can cause issues. If they get into the urinary tract, which is not very far from the digestive tract, they can cause UTIs. If they get into open wounds, they can cause wound infections, they can cause skin infections, they can get into bloodstream, they can cause heart infections or endocarditis. And the problem with a lot of Enterococcus bacteria is they are becoming resistant to a lot of our antibiotics. So they are getting harder to treat. We also do have some Streptococcus bacteria that have group C and G antigens on them. Again, these are normally found more in animals, and we sometimes pick them up. They can sometimes cause upper respiratory issues, kidney issues, pharyngitis, sore throat. They're not super common, but there's lots of those different Lancefield antigens. Now the A antigens, like what Streptococcus pyogenes is, when we're trying to diagnose to see if someone has Streptococcus pyogenes, and you may have had strep throat at some point in your life, well, they want to make sure we, one, diagnose it correctly so we can treat correctly. So they're usually going to do a rapid test. So if you've ever gone in for a throat culture, whether you've had strep throat or not, they usually do a swab. They leave the room, then they come back 20 minutes later and say, you have strep throat, or, oh, you don't. They can do a rapid test. They're looking for specific antigens that would react with antibodies in a test. It's one of those rapid, I'm going to say the rapid diagnostic tests. You get results in minutes. Otherwise, they're also going to they can grow up the bacteria in a lab, and they can start to look for hemolysis. Strep pyogenes is beta-hemolytic. Others can be as well. So then they start looking at what antibiotics is this bacteria resistant or sensitive to sort out which bacteria it actually is. Now, if you suffer from a streptococcal infection that has A or B antigens, generally penicillin is given. It's the top antibiotic. However, there's people that have had issues with strep bacteria. So they've had rheumatic fever, they have a recurrent strep throat. I've had students who are like, yeah, I get like 6 strep throats a year. Sometimes they do a prophylactic penicillin. It's a low dose of penicillin that you just take every day to try to keep this bacteria in check so you don't keep getting all those throat infections. Otherwise, the group D, the enterococcal bacteria, usually they're a little harder to treat. It usually requires some type of combined therapy. So you're going to have a combination of different antibiotics. Now, we haven't really talked about this oddball group. I like to call it the oddball group. It's called the Viridans group. It doesn't have any A antigens or B antigens or D or anything else. So it doesn't fit into the Landsfield classification. We still know genetically it's still a Streptococcus bacteria. It just doesn't have those antigens on its surface. So it's the oddball group, but we call it the Viridans group. There's a lot of Streptococcus in this group. Most of them don't cause a lot of issues. A lot of them are found in the mouth. We find them in the nasopharynx. We do find some in the genital tract, some on the skin. So most of them not invasive. The main time we usually have issues with these bacteria in the Viridan group, and of course I'll have an exception, but most of the time with this big list is during dental procedures if you were immunocompromised and you get some of those normal flora in your mouth into open wounds, because when you ever go to the dentist, you don't floss as much as you're supposed to, so when they floss, you bleed. That's an open wound that these bacteria could get into. And if you're immunocompromised, that could cause serious issues. It is another reason why they do a prophylaxis for those that are immunocompromised before dental visits. No, and I'm like, again, generally those that have pre-existing diseases or immunocompromised are going to suffer the worst because it can get into the bloodstream and cause bacteremia. It can cause meningitis. It can cause abdominal infections. It can cause tooth abscesses, but some of them can cause endocarditis as well. Again, normally the biggest group that's going to suffer those more severe side effects are the immunocompromised patients. However, there is a bacteria found in this group, because it doesn't have A antigens or B antigens or anything fancy, is Streptococcus pneumoniae. We call it the Pneumococcus, and it causes about 60 to 70% of all bacterial pneumonias. There's viral pneumonias too. But this particular bacteria causes about 60 to 70% of bacterial pneumonias. It is where it gets its name, but Streptococcus pneumoniae doesn't only cause pneumonia. Yes, it can cause pneumonia. It is in its name, but it's not the only thing it can cause. Now, this bacteria usually does like to hang out in pairs, sometimes in short chains. It likes chocolate agar or blood agar, and it usually likes a little bit more carbon dioxide when it grows. If we give it an all oxygen environment, it would actually die. So it likes more of a low oxygen, high carbon dioxide environment to grow best. So if we're gonna ID it, grow it in a lab, we gotta make it happy. Now, other than causing pneumonia, this bacteria also is one of the top causes of ear infections. This bacteria can get into the middle ear through the eustachian tube. So you can get some of those upper respiratory secretions, they can get in there, and it's a top cause of ear infections, especially in children. Now, where we find the bacteria, so a lot of people carry this as part of the normal flora, found up in the nasopharynx region. Now, 5 to 50% is a very big range. It can depend on location in the world, in the United States. It can depend on, do you work in healthcare or not. There's lots of different reasons that it can change, but it's part of the normal flora. Luckily, it doesn't survive very long outside of the body very well, but the people that are at highest risk, young children, elderly, those that are immunocompromised, and people that live really close to each other, because it's not spread easily because it doesn't live outside of its host for very long. So those that live in really close quarters with each other are going to be at a higher risk of picking it up. Now, when the bacteria gets in to the body, it does grow very, very quickly, which is going to induce an inflammatory response. Whether it's going to be a lot of pain, whether it's going to be fever, but it's going to introduce that inflammatory response. And yes, it gets into the middle ear and causes ear infections. So we can see how in a mic it can actually get in through the eustachian. Well, it causes the ear infection. People that this happens a lot actually have tubes put in. So if you've ever known anyone that has had a tube put in their ear, there it is, right there. Otherwise, this is an inflamed ear during an ear infection. And again, you've got lots of fluid in there. It's going to be painful. You're going to feel all the pressure. So tubes are put in to always keep the fluid that's in there draining. Otherwise, fluid in your ear is a great place for bacteria to grow and thrive. Now, when we diagnose some of these pneumococcal infections, so these are cocci-shaped bacteria that can cause pneumonia. Well, we can gram stain them. We can look to see gram-positive cocci. We can look to see that it likes to hang out in pairs and chains. But we can do some other tests as well. We can do something known as a Quelig test. This is looking to see if the capsule swells. We can look to see if it has alpha-hemolysis. We can also look to see if it's sensitive to the antibiotic optogen. And so we can actually see this little white paper, it's a little paper disc. It's soaked in the antibiotic optogen. I don't know why it's a P for optogen, it's the second letter. But we notice that this bacteria, it is a greenish color. That's an indication of alpha-hemolysis. But the bacteria doesn't grow anywhere near this antibiotic, which means it's sensitive to antibiotic. This bacteria is also soluble in bile. So if we put this bacteria in some bile, it would dissolve. Unlike Enterococcus bacteria. Entero means intestine. So Enterococcus bacteria, they're used to bile because that's where you find it. And so they're not soluble in bile. Your streptococcal bacteria, especially this one, would be soluble in bile. Treating it, penicillin, normally the top drug. However, we are getting resistant strains of these antibiotics. And we do have a couple vaccines for the Streptococcus pneumoniae. Now, we've got some that are usually for high-risk individuals that are usually for older. This is those that go off to the dorms. They usually recommend this as a vaccine because you're living in close quarters with other individuals. Those that go into the military because they're going to live in close quarters in the barracks, they're usually recommended to get this vaccine. And we also have a vaccine for children. And again, it can decrease the chance of pneumonia, but it decreases the chance of all those ear infections as well. So, my next little concept check question is bacterial pneumonia by Streptococcus pneumoniae, is it highly contagious? Now, Mike, it's false. You have to be in really close quarters with someone. So it doesn't survive for long periods of time outside of the body. So we consider this as false for being highly contagious. Now, we're going to move on to gram-negative cocci bacteria. So everything else we've talked about, all gram-positive, all would show up as purple in a lab after a gram stain. The next ones we're going to talk about are the gram-negative. They would show up as pink. I didn't want to put negative in pink. It might be hard to see, but they would show up as pink. Now, these bacteria are found in the mucous membranes of warm-blooded animals, including us. We've got several different bacteria genera that are gram-negative in cocci. There's only one big one that affects humans. It's rare for the other ones, so we don't talk about them as much because it's pretty rare. But Neisseria, the genera of the genus Neisseria, does have several different human pathogen species in it. And we're going to talk about the two most common ones, the Neisseria gonorrhoeae, it's got two E sounds in there, and Neisseria meningitis. Now, they are generally diplococci. They like to hang out in pairs. They are cocci, but they almost look a little oval. They kind of say bean-shaped. I don't know if I see bean, but they almost look squished when they hang out together in a cocci. They do have capsules. They do have a pili, so they can connect to each other. They don't survive very long outside the host. They are aerobic. Some are microaerophilic, meaning they like a little less oxygen than normal. They can produce the enzyme catalase, like the gram-positive staph bacteria. They can also make something called cytochrome oxidase, so we do an oxidase test to test for them. And they do require enriched complex media. We can't just grow them up on our normal plates. Now, our first Neisseria that we're going to talk about is Neisseria gonorrhoeae. We call it the Gonococcus bacteria, and it causes the disease gonorrhea. It's an STD. Now, some of the factors that contributed to its pathogenicity, which is, these are the virulence factors, is that it does have fimbriae that allows it to attach. So it allows it to stick, and it slows phagocytosis. It also makes an enzyme called IgA protease. This is an enzyme that cleaves or inactivates our immunoglobulin, our antibody A. So we make antibodies to target these different things and target them for destruction. Well, this bacteria makes an enzyme that stops that from happening. So it's getting trickier than our immune system. Now, luckily, this is a bacteria found only in humans, but gonorrhea is one of our top 5 STDs. The infectious dose, so how many bacteria it needs to cause disease, is somewhere between 100 and 1,000. That can depend on health of the host. But it doesn't generally survive any longer than an hour or 2 on surfaces. So it has to generally get spread host to host. Now, this is just looking at number of cases, and I'm like, luckily for us, we've seen a downward trend in the number of cases of gonorrhea. Yay! So we kind of saw a spike in the 70s and the 80s, and we haven't developed a vaccine. Mostly we are testing more, we're getting information out there, we're telling people how it's transmitted, and people are taking better precautions. However, my little note, and like this diagram's a little more updated, this one, we are starting to see a little upward peak. I don't know if it will continue or not, but I liked this because you could zoom in that, yes, there's cases around here. There's cases in La Crosse. This is a college town. There are cases of gonorrhea here in La Crosse. Now, I was going to say there's so many pictures out there for gonorrhea stuff. Now, diagnosing it, males and females generally have different signs and symptoms of gonorrhea. In males, they have what's known as urethritis. It's inflammation of the urethra. So there's going to be a yellowish discharge. It's a pus. It can cause scarring in the urethra. It can cause infertility. And 10% of males are asymptomatic, meaning they have no idea they have it at all, which means they could be spreading it without knowing they have it. In females, it can cause inflammation of the vagina. It can cause inflammation of the urethra. It can cause pelvic inflammatory disease, abdominal infection. And because it can cause damage and inflammation in the reproductive tract, it is a common cause of sterility as well as ectopic pregnancies. And 50% of females are asymptomatic, which means this could all be happening and you would have no idea whatsoever, which means it can be left untreated and it can cause that sterility or ectopic pregnancies. Now, some, kind of how we get it. Because it's not only causing STDs. And it's still a bacteria, it's out there. Anal intercourse can lead to proctitis. Oral sex can lead to pharyngitis and gingivitis. If you've got poor hygiene, if the bacteria gets anywhere on your hands and you touch your eyes, you can suffer from a very serious form of conjunctivitis. It can get into the bloodstream. It can travel throughout the body. It can affect the joints. It can affect the skin, causing a rash. And it can cause meningitis and endocarditis. Again, it just depends if it spreads. It can also depend on the health of the immune system as well of the patient. Now, if mom has this bacteria infection, and again, 50% don't even know, during pregnancy, infants can pick it up during childbirth, and it can cause a very serious conjunctivitis. It causes that eye inflammation, and it can lead to blindness in newborns. Again, it can get past just the eyes. It can cause respiratory tract infections, and so a lot of times infants are given antibiotics into the eyes right after childbirth. So if you ever wondered why they put antibiotics and goopy stuff all over the eyes of infants, they don't always test mom for this particular bacteria. So they just put antibiotics in the eyes to try to decrease that chance. Now, diagnosing it, well, we're going to gram stain it. We know it's gram negative, so it's going to show up pink. We know it's cocci shaped, and we know they like to hang out in pairs. We also know they like to hang out inside of neutrophils. We know that some of our neutrophils like to eat them, but these bacteria figured out a way to not get broken down. And so underneath the microscope, we can actually see some of these bacteria inside of our white blood cells. That's a huge diagnostic tool right there. There's not a lot of gram-negative cocci-shaped bacteria out there, especially not ones that like to hang out in pairs and hang out inside of white blood cells. Now, treating it, most are given some type of penicillin or tetracycline. Depending on how it's working, you might be given a combined therapy. But a lot of times, if you've had one Neisseria infection, you're more likely to get another one. This is showing, again, some of the neutrophils with those gram-negative cocci in pairs all over. Now, we've talked a little bit about some of these virulence factors in some other bacteria, but we're going to talk about one more bacteria, our kind of last bacteria. It's Neisseria, so it's still gram-negative, it's still cocci, but it's Neisseria meningitis. It's called the Meningococcus. Now, it also has a capsule. It has fimbriae to allow it to attach. It has the enzyme IgA to break apart or destroy or inactivate our antibody or immunoglobulin A, and it has toxins. Now, based on its name, Neisseria meningitis, it can cause meningitis. However, it can get into other parts of the body and cause other things other than meningitis. Now, in humans, a lot of humans are just carriers of this bacteria. It hangs out in the nasopharynx. Those that live in close quarters with each other, in daycares, in dorms, military barracks, any of those are going to be at higher risk of picking up this particular bacteria. And so when it's picked up, it can enter the bloodstream and it can cross that blood-brain barrier, affect the meninges, and cause that meningitis. Now, generally, the symptoms are very, very fast, usually within 24 hours. Usually is going to cause, severe headaches. You're going to have a severe fever. You might actually have blood vessels bursting. You might have blotchy skin. And I'm like, it causes severe neurological symptoms, and it can be deadly because of that. Again, it's getting into that the meninge fluid, the cerebrospinal fluid, in the meninges, and it's causing inflammation. That really is putting pressure on the brain. Now, the toxins, again, it can lead to hemorrhaging. You usually end up with blotchy red skin everywhere. It can cause coagulation, vascular damage, breakdown and destruction of tissue. And you end up with this, it's called petechia. It's red spots that eventually turn black because of death of tissues that end up on the body. So yes, the pictures, it's horrific. It's death of tissues because this bacteria is destroying not just the nervous system, it's destroying the cardiovascular system and the blood vessels themselves. So when we diagnose it, we're going to gram stain some of that cerebrospinal fluid. We're going to look for those gram-negative cocci in pairs, in the fluid, in some of our neutrophils. We can also gram stain samples from blood or the nasopharyngeal sample. We can grow it up. We can run various tests. Mostly they're going to do a gram stain, and they're going to be looking for that fast result because you can get a gram stain in results in 10 minutes or less. Now, treatment, usually IV at that point. If you've got symptoms, you need to get antibiotics in your body ASAP. So treat it with penicillin through an IV. We also, cephalosporin can also work. And usually if you're diagnosed with the Neisseria meningitis, they're going to give antibiotics to everyone you've been in close contact with. So any medical personnel that has been working on you, family members of yours, any children that you might have had contact with, are all going to be given antibiotics because it can work so fast and because it can be deadly. Now, we have a vaccine for it as well. And so those that usually go into dorms that are at close contact with each other's military barracks are usually the ones that are advised to get the vaccine. Now, my last little concept check on here. A common cause of meningitis in young adults is from Neisseria. It's the Neisseria meningitis. So, this chapter we got through a lot of gram-positive cocci and gram-negative cocci. So everything cocci-shaped, lots of different diseases that can happen.